Hello and welcome to my Mitosis Lab video. I've brought back Etta and Malin today so they can be my spindle once again. And let's just hop right into mitosis. Before getting into mitosis, I first want to talk about the cell cycle. So the cell cycle has two, sometimes three parts. The two parts are interphase and mitosis, and sometimes the third one is cytokinesis. Otherwise, that's sometimes paired with mitosis. And interphase is actually broken up into three different groups. There's G1, S, and G2. And S phase is when DNA replicates, and so the chromosomes will duplicate, as we can see here on screen right now. So now I want to describe a little bit of what we're seeing on screen. So for our chromosomes, I have rolled up paper, and to show the three different pairs, I've done three colors, and they're also three different sizes. So we have the big pink ones, the medium green ones, and the small blue ones. They also all have alleles at the end of their little chromatids. So the black lines on them represent the different alleles. And for the centromere, which holds the duplicated chromosomes together, I have some black ribbon. Actually, it's black yarn. Oops. So now let's get into the process of mitosis and the three main reasons of why a cell goes through mitosis. The first is so that the cells can replace old or dead cells. Another is growth, so if somebody's growing, their cells will divide and therefore go through mitosis, or repair, to repair some damaged cells. The first stage of mitosis is prophase, and in prophase, three important things happen. 1. The chromosomes condense. 2. Nuclear membrane disappears. And 3. The spindle forms. Next we have metaphase, and in metaphase the chromosomes line up end to end, and they randomly line up, and they don't have to line up with their homologous pairs. They can just line up wherever, as long as they are lined up. In anaphase, the spindle are going to grab the chromosomes, and they are going to unduplicate, so the centromere is going to dissipate, and the sister chromatids are going to separate. Then the nuclear membrane of the cell will start to pinch together to form two new cells. So that brings us right into telophase and cytokinesis. Now we have our two new cells, so here's one, and here's the other one. They are going to look identical to the parent cell, unless of course a mutation happens, but we're not talking about that right now. And just to be clear, telophase is the separation of the chromosomes and the chromosome splitting, and cytokinesis is all of the other stuff, so the organelles and the mitochondria and what have you. Okay, so that's all I have for my Mitosis Lab. Thank you for watching, and I hope that you learned something, although I'm sure you didn't, because you're my professor, and you taught me this. So I'd be surprised if you learned something. But anyway, I will be putting my points of confusion in a different document, which I will be uploading to Canvas. So, thanks again. Bye.